Hi and welcome to my OCRA A-Level Biology revision session with me, Christine. So today's lesson I'm going to look at factors affecting enzyme activity. So let's take a step back and just remind ourselves that enzymes are globular proteins. They act as a biological catalyst, and they do that because they form temporary bonds with the substrate when they collide, which puts a strain on the bonds within the substrate. And what that does is that lowers the activation energy. So by lowering that activation energy, what's going to happen is that enzyme is going to cause the reaction to start in a much faster way. So that's why we call them biological catalysts, because they speed up the rate in which the reaction occurs. So if we were to look at the substrate concentration and we increase the substrate concentration, we know that as we increase that substrate concentration, we're going to end up with a higher collision rate. Therefore, if there's more collisions, there would be an expected increase in the rate of the reaction. And that's because if there's more collisions, there are going to be more enzyme substrate complexes being formed, therefore putting the strain on the substrate, allowing for that lowering of the activation energy and the reaction occurring. So when we are at a lower substrate concentration, what we're saying is that some of the active sites are actually free. Not all of the enzymes are being utilized. And that therefore means that the rate of reaction is not yet at what's known as its Vmax, the maximum rate of the reaction. So as we increase the substrate concentration, what we can see is that there is quite clearly a curve to a point where we reach that maximum point. And that maximum point is where we have all our active sites becoming filled. So if all the active sites are filled, then that is the maximum rate of the reaction. So it doesn't matter how much more substrate I put in, it will never get past that Vmax, that maximum rate of the reaction, because the enzyme needs to process the substrate, converting it into the products and releasing it before it can accept another substrate in for the reaction to happen again. So once that Vmax has been reached, that is the point in which all the active sites have been filled and the enzymes are working at their maximum rate. So, well, what happens then if we change the temperature? So if we know that our substrate concentration increases is going to get us to our Vmax, what we can then do is say, well, how does temperature affect the rate of reaction? We know that our substrate have kinetic energy. We know that our enzymes have got kinetic energy. We know they're going to collide, and so therefore that kinetic energy is important. So as we increase temperature, we will see this very specific set of results which give us this curve in our graph. As we increase the rate of the reaction, so as we increase the temperature, what we will see is that there will be an increased kinetic energy. That thermal energy is converted into kinetic energy. If there's more kinetic energy, you're going to have more frequent successful collisions between the substrate and the enzyme. Therefore, you're going to have more enzyme substrate complexes forming. Now, it's important to know I have written ESC here as an abbreviation for enzyme substrate complex. So when I am writing my revision notes, I try to minimize the amount of words that I use and write it in abbreviations or little hints to myself. Please be careful. In the exam, you should write it as enzyme substrate complex at least once before you do any type of abbreviation. Okay, so I've increased my kinetic energy because I've increased my temperature. I've therefore got more successful collisions, more enzyme substrate complexes forming. Therefore, my rate of reaction has increased. Well, I've said here that the rate doubles every 10 degrees. What you need to know is that's known as your temperature coefficient, your Q10. So the increase in the rate of reaction with a 10 degrees C increase is known as your temperature coefficient. We say that the rate normally doubles every 10 degrees C, but what they like to do in the exam is actually give you a graph to pull that information from. So they will expect you to be able to interpret graphs and to work out your Q10. So if I tell you that 20 degrees C on my graph, my rate of reaction is 0.009, and at 30 degrees C, my rate of reaction is 0.017, what I can see is that my rate is different at the lower temperature compared to the higher temperature. So to work out my Q10, I take my highest temperature, R2, and divide that by my lowest temperature, R1, and that will give me my Q10 value, which is 1.88 reoccurring. So do make sure when you are looking at this that you note that although I'm saying that the rate doubles every 10 degrees C, and the textbooks will tell you these set numbers, they do expect you to be able to apply this knowledge to the question that's given to you. So you simply have to read the numbers from the graph and then input those numbers into the equation. And that equation is one that you should know. So as the temperature increases, my rate is increasing up into a point which is known as the optimum temperature. So when we look at humans, for example, our optimum temperature is our body temperature. So it's about 37 degrees C. But if we were looking at thermophilic bacteria, for example, they have a much higher optimum temperature. Therefore, their rate of reaction will continue to increase until their optimum temperature has been reached, which is about 65 degrees C. So it's again important to note that not all enzymes have the same optimum temperature. It depends on the enzyme and the organism that it comes from. But if we surpass the optimum temperature, if we go too high and we keep increasing the temperature, what will happen is what's known as denaturation. If we're increasing the temperature, we're increasing the kinetic energy more, therefore we're increasing the vibrations in between the bonds which are holding the tertiary structure of the enzyme together. Now remember, the enzyme has a very specific shape in its active site which complements the substrate that it's going to work on. If we are changing the shape of the active site because we're breaking the bonds in the tertiary structure, the enzyme is no longer going to be able to fit with the substrate. It won't put any strain on the substrate and therefore will not lower the activation energy. So it therefore doesn't work. The rate of reaction stops working, and you can see that that is a decline in my graph. Now, thermal denaturation is irreversible. I like to think this through in if I was cooking an egg. Egg is made up of protein, the albumin, the white is the protein. If I were to cook my egg, I can't get it back to being raw. It is irreversible. Once the temperature has been risen to a high enough level that you have thermal denaturation, you cannot get it back. It is irreversible. Well, pH is another condition which is going to affect the activity of the enzyme. Now, pH is determined by the availability of hydrogen ions. Potential for hydrogen is what pH stands for. So we know that if we have a low pH, there are lots of hydrogen ions, and if we have a high pH, there are very few hydrogen ions. So different enzymes are going to have different optimums, and we know that hydrogen ions interact with the polar and charged R groups. Now, because they interact with these polar and charged R groups, they're going to affect the tertiary structure of the enzyme. Remember, the tertiary structure needs to be specific so that the active site is able to form the bonds with the substrate. So if we move away from the optimum, whether we move too low or too high away, we are going to affect that tertiary structure. We're going to affect the way in which the enzyme and the substrate can form those bonds. So therefore, the activity is going to decrease. The rate of reaction is going to decrease. Well, the good thing is that with pH, it is a reversible denaturation. As long as it doesn't go too far away from the optimum, it can make its way back to that optimum by having some way of buffering the pH. So whenever we do an enzyme activity and we're looking at an investigation, we tend to use a pH buffer. So pH buffers are really important to maintain the structural shape of the enzyme or the protein, because remember, an enzyme is a globular protein. So pH needs to be maintained so that the enzymes, the protein, can function at its optimum. So if we take a step back and look at our protease then, protease we know is an enzyme that's going to digest our protein down into smaller peptides, and then other proteases will break down smaller peptides into amino acids. Well, it's important to note that trypsin is an enzyme, a protease enzyme that is produced by the pancreas, and it will do its digesting inside the small intestine. Well, therefore, its optimum pH, as we saw in the graph before, is around pH 7. Well, when we eat the food in our diet, what we do is the food will go down our esophagus into our stomach. Now, to ensure that any pathogens that may have been ingested are stopped or at least slowed down, what we have inside our stomach is we have stomach acid. Well, in that case, we produce a different type of protein that has a different optimum with regards to its pH. And this is known as pepsin. So as you saw in the graph before, pepsin's optimum pH was a pH of 2. Well, the cells that produce the enzyme, which is going to produce pepsin, actually produce it in an inactive form, what's known as a precursor enzyme. So it's called pepsinogen when it is inside the cell and in its inactive form. After it has been released, exocytosis has occurred. It has been released out of the cells in the stomach into the stomach environment, which is an acidic environment. The acidic pH of the stomach will actually change that tertiary structure, and that makes the pepsinogen be converted into the active form, which is pepsin. That is an important step in the process because that ensures that the pepsin, the active form, is not within the cells. Because if it is within your actual cells, it would start to digest the cells proteins, whether that's the proteins in the membrane or whether that's the proteins in the cytoplasm, but it will actually digest the cell's proteins and therefore cause tissue damage. So by producing it in an inactive form, a precursor enzyme that is only activated when the pH is correct, that means that it can ensure that there is no damage within the tissue. Now, you'll learn more on this when you do Module 6, so do check out my video on Module 6 looking at gene control. So, how do we know this? How do we know that temperature and pH have an effect on enzyme activity? Well, we do this through using investigations. So, if we're looking at investigations then, we're going to start off with one that you will need to know, which links to both Module 2 enzymes and Module 5, which is part of excretion and liver. So hydrogen peroxide is a toxic metabolic waste product. Now, catalase is an enzyme which is found in both plants and animals because they do metabolic processes. Therefore, they both produce hydrogen peroxide as this toxic metabolic waste product. So catalase is this enzyme that is going to break down the hydrogen peroxide into the less toxic, more safe substance, water and oxygen. So if we understand that an enzyme called catalase can break down this toxic substance into water and oxygen, what we can do is we can actually do an investigation on this. So as you can see, I have a hydrogen peroxide solution. I have a liver, which is going to contain my catalase. I have this in a conical flask, and then I have a delivery tube going to a measuring cylinder of water, which is upside down, and I would be able to measure the volume of gas collected. In this case, because we're talking about breaking down hydrogen peroxide, the volume of gas being collected is oxygen. So I can use liver, or I could use potato, for example. So they do like to give you practical where it's either about liver or it's about the potato. So let's look at some practical skills then. So if I have an experiment which is going to investigate the effect of a substrate concentration on the rate of an enzyme controlled reaction, I'm going to use this setup. Now, the first thing I need to understand is, well, what's my independent variable? Well, I was already told it was substrate concentration. So therefore, I need to ensure that I have different concentrations of hydrogen peroxide. So in this case, I've got 5 different values for my independent variable: 2, 4, 8, 16, and 20 arbitrary units of hydrogen peroxide. So they're the things that I'm going to change as the investigator. I am going to do a serodilution. Do check out my video on how to do a serodilution to get that concentration. Then what I'm going to do is I'm going to measure my dependent variable. So my dependent variable is the variable that I'm going to measure. In this case, it's the rate of the reaction. And I'm going to measure that based on the volume of gas that's given off every 30 seconds for 3 minutes. Now, with any investigation, it's always important you note your independent variable, your dependent variable, and your control variables. So the control variables are every other variable that you must keep the same so that you can ensure that the investigation isn't affected by any of these other factors. So as we discussed, temperature and pH will affect the rate of the reaction. So therefore, we want to control the pH using a buffer solution. We want to control the temperature. We would use a water bath. We obviously want to control the enzyme concentration. So to do that, we may have the enzyme specifically isolated, or if we're using, for example, liver or potato, we would want to have the mass of that catalase source controlled. And obviously, if we're changing the concentration, we want to control the volume of the hydrogen peroxide, that substrate. So it is important that you know your variables and you know how to switch them around. So if I change this investigation to look at the effect of temperature, my independent variable would therefore be temperature, and concentration of the hydrogen peroxide would become a control variable. So these are always the skills whenever you plan out an investigation that you need to consider. Then you always want to make sure you do repeats at least 5 times to calculate a mean and standard deviation. Now, some textbooks will say it's 3 times. You can do 3 repeats and that will be enough to calculate a mean. However, standard deviation, you should really have 5 repeats. Once you've got your mean and standard deviation, what you would then do is graph out the data and conduct a statistical test. So do check out my video on statistical tests. So the last thing I want to talk about when it comes to factors that affect the activity of an enzyme is what's known as inhibitors. So a competitive inhibitor is something that is similar in shape to the active site. And because it's similar in shape to the active site, what it can do is it can actually form those bonds and therefore block the active site temporarily. So when the substrate comes in and tries to collide and bind with the enzyme active site, something's in the way, it cannot bind, and therefore the rate of the reaction would be lowered. Another type of inhibitor is known as a non-competitive inhibitor. It's a non-competitive inhibitor because this time it's not competing for the active site. Instead, what's happening is this non-competitive inhibitor is going to bind to an allosteric site. When it binds to the allosteric site, it is going to change the tertiary shape of the enzyme. So the tertiary structure, the active site, is going to change. It's therefore no longer complementary. It can therefore no longer catalyze the reaction. So when we look at the rate of reaction, if we are increasing our substrate concentration, we know that the rate of reaction will continue until it reaches the Vmax. So if there's no inhibitor present, we will get to what's known as our Vmax that we discussed before. Well, if we have a competitive inhibitor, that is obviously going to decrease the rate. Remember, this competitive inhibitor is competing for the active site. So if the competitive inhibitor collides and binds to the enzyme before the substrate, it is therefore going to block the substrate and decrease the rate. However, if we increase the substrate concentration, what you will find with a competitive inhibitor, the competitive inhibitor will still result in you reaching your Vmax, because when you have more substrate concentration, they are going to be able to outcompete the competitive inhibitor. Now, that's only if the competitive inhibitor is a fixed volume. So if there is a fixed volume of this competitive inhibitor, the more we increase the substrate concentration, we can overcome that inhibition and reach that Vmax. So basically, when you look at the graph, if the lines get up to the top where it plateaus, that indicates it's a competitive inhibitor which was decreasing the rate but was overcome by an increase in substrate concentration. Whereas if we have a non-competitive inhibitor, again, we have a fixed volume of this non-competitive inhibitor, that will decrease the rate and the Vmax will not be reached. And that will not be reached because obviously the non-competitive inhibitor has bound to the allosteric site, changing the active site shape. We therefore have less active sites available to do the reaction, so we will never reach that Vmax point. So what they love to do in the exam is give you a graph and expect you to interpret it. So it's important you note if your line follows the same path but is lower down and never reaches the Vmax, that is your non-competitive inhibitor. Well, another process which actually inhibits the rate at which a product is made is actually called the end product inhibition. So end product inhibition is a way in which the cells can regulate the rate of a metabolic reaction. Basically, they want to prevent the overproduction of a final product. If you think about it in the sense of, if you're making too much of a product, for example, you are writing letters and putting them in envelopes and you need to post them. If you end up with far too many envelopes to post and you have to therefore wait, you don't want more letters being printed off. You want to process and post the letters that you have already made, and only once they've been processed do you want to have the printer printing more letters out, and then you can put them together and post them off. So cells work in the same way. They don't want to have this overproduction, because if they have an overproduction, it can affect the way in which the cells can function. So instead, what happens is the product, the end product of the reaction, can act as an inhibitor to the enzymes that are going to produce it. This ensures you're not wasting resources. It ensures there's not too much, this overproduction of the end product. So therefore, in this case, you will learn about oxaloacetate and the fact that there are lots of intermediates that are necessary to result in you being able to produce ATP as part of the Krebs cycle. Well, there is an end product called lysine. If there is too much lysine, what the cell will do is the cell will actually inhibit enzymes that are going to produce lysine. So there is an inhibition where the lysine can bind to the allosteric site of two of the enzymes that are necessary in the pathway to produce itself. So with any enzyme, you always have the substrate and the product. So if we look at the first enzyme, my substrate is oxaloacetate, my product is intermediate 1. If we look at enzyme 2, my intermediate 1 is my substrate, my product is intermediate 2, and so forth. And each one, the substrate is converted into the product by the enzyme catalyzing the reaction. Well, if we inhibit the production of intermediate 1, we can stop lysine being produced. If we inhibit intermediate 3, we can stop intermediate 4 being produced and therefore inhibit lysine, that end product. So when they give this to you as a question, they like to throw you off by giving examples that you've never heard of before. This is quite a common example, but they tend to make it more complicated by showing you other pathways. What's important for you to note is the end product needs to inhibit itself being produced. That is the correct pathway for an end product inhibition, okay? So what we say is that it is a non-competitive because it can bind to the allosteric site, but it is a reversible inhibition because when we have processed all those letters, when we've posted them off, we will then start the printing again. In the case of when the lysine concentration has dropped, then oxaloacetate can start in the reaction again and be broken down to end up with that end product. So it is a non-competitive because we're attaching to the allosteric site and changing the tertiary structure, therefore changing the active site shape, but it is reversible because it will be removed when the concentration gets to a level where the cell can manage it again. So I hope you've liked this video, and if you have, then please do click the like button and subscribe to my channel. Also do check out my revision platform, www.aiqchat.com.